then I think we'll just get started with reusable data analysis. The main idea is that we want to address how live coding with instant outputs is beneficial for being fair. I want to show you how to run and adapt existing Jupyter notebooks and run code in them. And we're trying to show you the benefits of these types of computational notebooks for analysis, show you how to use one, and kind of discuss how these notebooks can help us be more fair. And so before we begin, I'd like to do a brief little survey of what our pre-existing knowledge is with coding and notebooks, because I just want to put a disclaimer out there. There's absolutely no coding experience necessary for today, but you will be looking at some code. So I thought it would just be nice if we had a little survey of who's familiar with what. So we have this little poll in our shared notes. And maybe you can just add a little +1 at the end of each statement if it applies to you. And then we'll get a little bit of a feel for the experience in the room. Okay, so I'm seeing some data analysis before. That's awesome. That matches with what we discussed yesterday, I suppose. And you've used R or Python before. That's great. So you will not be too surprised when we look at a Jupyter notebook. Okay, it seems like people are not using Excel for data analysis. That's great. All right, well, with that, we'll jump back into these slides. So I probably will be preaching to the choir here when I tell you that data analysis and research are a very iterative process. We're used to collecting data, preliminarily analyzing it, and then that will inform our next steps in experiments, or we will modify our experiments and then collect more data and then analyze it again. So that kind of iterative process requires us to analyze data over and over and over again and potentially only make slight alterations. And so we could very easily rely on previously written data analysis that we have produced. And yeah, so obviously this will be quite a challenge if we're not documenting our data analysis process properly, and we have to reinvent the wheel of analyzing data over and over again. And so this is what we call ad hoc analysis. If every time we're getting data, we're writing new data analysis to analyze them, that's ad hoc. And yeah, and then hopefully we'll go through some iterations, and then we'll arrive at a final version of our results and our graphs. And then ideally, we have documented every step of our iterations, so we know exactly where we started, what we tried, what worked and didn't work, and how we arrived at the final versions. And so plotting in R or Python is like a very natural starting point to learning programming that allows you to create more professional plots than those available in Excel. And from what I'm seeing in our poll, this is a step that most of you have already taken. So that's really great. And another great advantage of R or Python is that it's a lot easier to recreate those plots because you actually have the code written in front of you instead of just clicking boxes and setting check marks to kind of get a plot, and then trying to figure out how you did this the last time. And I should mention this here as well, but there are many coding resources out there, or learning resources for learning R or Python, including other software carpentries courses and Bio-IT courses, if that's something you're interested in. So we talked about electronic lab notebooks yesterday, and how they're vital in the process of verification. And so today we want to talk about computational notebooks, which are essentially the equivalent to these lab notebooks, where you can store your data, your analysis, you can document how you got through the analysis, how you generated the code, etc. And so one example of these computational notebooks is Jupyter notebooks. So maybe just a show of hands, who has used a Jupyter notebook before? And if not, have you used any other notebooks before? Okay, right, I'm seeing some familiarity. That's great. Does anyone want to chime in and talk about other notebooks, computational notebooks they've used before? I think another one would be these R notebooks, or these R Markdown documents. That would be very similar alternative. These computational notebooks are essentially the lab notebooks for scientific computing. And instead of pasting DNA gels or lab protocols, we can embed code and data and then text to document the computational methods we used. And we're going to kind of look at this using Jupyter notebooks. So Jupyter notebooks are interactive web applications which allow you to type and edit lines of code in different programming languages. The name Jupyter comes from 3 of them, Julia, Python, and R, as far as I'm aware of. But I think the software can actually accommodate like well over 20 coding languages at this point. And yeah, it allows you to view the output of your code immediately, which is a huge benefit compared to other ways of documenting your code. Yeah, and so these Jupyter notebooks run on a so-called kernel, which is just a computer program that executes and runs the code for you. And this can either be run locally on your computer or on a server. And what is really nice is that these notebooks can be exported as PDFs and HTML files, which are really graphically appealing already. Like you can make pretty fully formed documents with them and share them. You can upload them to GitHub, and GitHub can actually render them itself too. So these are some of these examples. Yeah, so some of the things that should go into your notebooks to keep them fair is your motivation, the experimental setup, how you got the final results, your input data, parameters, methods, and then you kind of want to record your intermittent results, what you concluded out of them, and then modify your code and, you know, keep track of your iterations, your outputs, write down your conclusions. One thing to keep in mind, these notebooks are only as good as your self-discipline. So there are a couple of things we need to keep in mind. So these notebooks have to be shipped with all of the input files. So if you're reading in a table or a CSV file or maybe an image, you will have to ship these images or these files with the notebook if you want to share them. And we'll look at this later. We have a minimal notebook prepared for you, and it will come with an image, it will come with a data file. And if you don't share these along with the notebook won't be functional. And another thing that is important, and I actually stumbled over a little bit in preparation of this, is the environment that you're running it in. So what are the computational resources that this notebook needs? And that can be something like the amount of storage space you need, or how big the data is that you're shipping with it, but also potentially which packages you've been using to analyze your data and which packages somebody else would have to install to be able to recreate your data analysis. And these are things that I've said before, but use this notebook to document your decision process, to document the parameters and what they mean, and continuously try to decide what you need to retain in your document and what you need to clean. And then finally, follow some good coding practices. And for that, I'm going to go over a couple of examples. Yeah, so notebooks really shine in orchestrating short data analysis, like step-by-step operations. They're good for capturing parameters, adding interpretations, and kind of acting as a user interface. What they're not are tools to create really complex, really long pipelines. So if you want to write a pipeline, let's say from sequence alignment to annotation to differential gene expression to gene ontology analysis or something, then you're chaining so many big pieces of software together that are not quite made to fit inside a Jupyter notebook. There's a lot of other tools that you can use to do this, and we can chat about them at the end of this section. Yeah, this is what I just said. They're not really made to run really, really long, computationally intensive programs. So let's go over some good coding practices, which might be relevant, especially since a lot of you have already used coding and have some experience. Using coding standards is really helpful in being able to understand other people's code or have other people understand your code. It's going to make it much more easily for you to reuse that code if you go back to it. It's going to make it easier to read in general and to try and detect bugs and errors, and will most likely also generate results faster. One thing that we like to do in our lab for this purpose is have code reviews. So each week we share a little chunk of our code with our lab members, and we have them look over it and kind of see what's counterintuitive, what we don't understand, what seems to go against our coding good practices. And so that really opens our eye to those things that are non-intuitive to other code readers. Some of these best practices are to write as few lines as possible, because this will make your code more human readable and also faster. Really important is to use appropriate names to describe your variables. Don't use df for data frame, or don't use XYZ for all of your variables. Don't use test1, test2, test3. Those are really hard names to keep track of, so try to be more descriptive. And then segment blocks of your code that go together. So if you're loading in a file, make that one block. If you're creating a plot, make that a second block, and like don't have them bleed into each other. It's going to be much more easy to repeat. And try not to repeat yourself. If there is a task that you find yourself coding over and over and over again, it's a good indicator that this can be done with a function. So either write a function yourself so that you can do this over and over again. For example, if you're always reading in a very specific file that needs like a lot of parameters, then maybe you want to write a function to import these types of files. Or most likely there are already functions out there that do that for you. There are plenty of R and Python data wrangling packages available with a plethora of functions. So here I have some examples for you. So one of them here at the top, this would be a less good example of a chunk of code, and the bottom would be a much better example. And maybe people want to just type in the chat or type in the pad or speak up what the differences are that you can see in this piece of code and what you like about it and what you don't like about it. So the one big difference between these two chunks is the separation of the individual steps that you're performing. So like you were saying, you might not even know what every single line of code does, but you can understand that they're different things. And it'll make it much easier for you to maybe look into, oh, I don't know what factor does. Let me look that up. But you know that it's independent of the plotting, for example, because that's sectioned off. I think that's just an oversight. You're correct. In the second box, you would definitely need to load the package as well. Yeah, that's correct. So it's better visible, but it's actually, it would have a bug in it if you weren't loading the library. Good eye. One of the other things that we'll look at in a second is using indentations to mark beginning and ending of structures and to avoid long horizontal blocks, because they're really difficult to read for the human eye and you might have to scroll left and right. So there's an example here. We're just looking at the piece of code that is doing the plotting now. And so what you can see here in the top chunk, Everything is aligned at the very front, but actually this is technically one line of code. So in the second chunk, we're using some indentation. For example, this is much further indented because this is still part of the initial ggplot function. And then within ggplot, it's customary to, with using the add addition sign to add different layers. And so each of those layers will get their own line. And since this layer is a very long command, we'll break that off again and indent it even more. And this is probably the most important part. Leave comments on what your code does and leave other people comments on what your code does. So for example, before you start the whole ggplot function, you'll insert a little comment. In R, you can do that with the little hashtag symbols, and then this piece of code won't be executed. So this is really ideal to leave comments for yourself. So here's somebody who commented plot the graph. This line is subsetting the SD from the light condition column. Here we're coloring by genotype. This line defines the plot title as well as the title of X and Y axis. And then this line changes the colors of the groups. And so now, even if we're not used to writing R code, we can at least kind of appreciate what each line of code does. And so with this, we're going to jump into our first exercise with the Jupyter Notebook. So just a reminder, we're doing this exercise 1A. You want to click or look at this screenshot right here, which labels a couple of different parts of the anatomy of this Jupyter Notebook. I think the screenshot is taken in a local installation of the Jupyter Hub. And then just go ahead and fill in what you think these different parts are, or note down if you can't identify them in what you're looking at. That's totally fine too. And work on changing the name, saving the notebook, and then try running some of the code chunks and go ahead and write down what you can observe. So I will share my screen and kind of walk through the exercise with you. I am using the EMBL Jupyter Hub for now. So as you can see in my Arabidopsis folder, I have all the files. I made a duplicate or a copy of the student notebook, light conditions. And I opened both of them, and they're running right here next to each other. So what are the different files? Exactly. So A, like you identified, is just the name of the notebook or the name of the file. B and C are toolbars of different ones. This is the more in detail toolbar. C is kind of the toolbar for the most used options. You can see that they're arranged here a little bit different. We have this detailed toolbar a little bit more top left, and then our most commonly used functions directly above the notebook. Then we have D. D is a little drop-down menu. We have that here as well. It allows us to select the type of cell that we'll be running, whether it'll be code or markdown, for example. And then we have too many tabs open, clearly. And then we have E, F, and G, and there are examples of a so-called cell. So a cell is just a chunk of code in this notebook. So up here, I have this light blue bar. This marks that something is highlighted. So if I click into it, I can see the cell itself, and I can see that it's set to markdown. And so now I can use markdown syntax to write in it. So for example, we're using this to type the title. I can also use this to type a subtitle in and then hit run. And so it will execute it. But as Chiara mentioned, this is a markdown cell. So all it's doing is generating the text for me. It's not running any code. The same would happen if I highlighted this. Oops. and clicked on run twice, it's just going to run the next chunk with it, which is also a markdown. Okay, so then what do we have to do next? Okay, so changing the title of the notebook and saving it. So changing the title, this one you can change here, as I showed you. You can add a subtitle. If you wanted to change the name, you would do that over here and select rename. Then you could, for example, add your initials to it to indicate that you've been working on it. And if you change something here, then you can run this, and then you can hit the little disk icon to save it. Okay, so I think that was this so far, and now we want to run all code. Okay, so in the top toolbar, we click cell run all. Let's see if I can even find that. I think that's this one here. So no, do it. Okay, so my kernel actually wasn't running, which you can see at this little, so we're running an R kernel, and up here we have this little circle, which currently here is empty, meaning the kernel isn't actually running. And so now here it's filled in. So now my R kernel is running and ready to run some code. And so I can use the run button to actually run the code. So there is a, if we're going about executing the next couple of chunks, there's a slight difference between people using the EMBL one and the band one. If you're using EMBL, the library that we're using, ggplot, is already installed, and you're more than welcome to just start executing code chunks down here. If you're using band, unfortunately, ggplot is not installed, and you will have to do that. So please do execute the install packages chunk using the little run symbol, and then it will install it, which will take a moment. And so I think in the meantime, we can start the next exercise. Okay, then I'll start with my run through so we can compare outputs. So again, we're in the Jupyter EMBL tab right now. And let's see what, okay, so first thing, we want to change the author's name. So I'm scrolling up. The author is in this cell, it's highlighted light blue. I'll double click to open it. And now I can just add my name in here, hit run again, and now my name is here in the text. So now we want to add a new cell to add some details about the file that we're loading. Yeah, so we can use the plus to add new cells. So I'll activate this one and hit plus to add in a new one. And so now we're supposed to write some more text in here, which I'm copying from the pad and pasting it in here. And so this is one example of what I think is a too long horizontal block, because I have a scroll bar here despite having a really big screen. So I would probably try to add some line breaks here. So one mistake that I made now is that I hit run and I'm getting an error. And that is because I selected the cell to be code, but what I really wanted is markdown. So I'm changing it up here. And then I can hit run again. And so now I have plain text appearing. There's also keyboard shortcuts for this. You don't have to use this run icon. I use Control and Enter. And that will do the same thing. There's also keyboard shortcuts for, I think it's Command and Enter, and that will execute that chunk and also add a new chunk, which can be convenient. Okay, so we ran that. Okay, and the same can be done. We can also remove cells. So if it's highlighted, I can use this little scissors icon and just remove the cell again. But we can also undo this with edit, undo. That's working great for me. And then we'll have it back. Okay, so in our next step, we're supposed to change the colors of our graph. So we're going to our visualized data. And since our colleague annotated this really well for us, we know that this chunk is for plotting the graph. And we know that this line is where we change the color. So we have hex codes in here, which are the very programmatic way of selecting colors, but we can also absolutely use strings. So for example, I can just use the string green, blue, and red, for example. And then I can execute this again. And then I have changed the colors on this plot. So finally, we want to save the graph under a new name to add our initials to the file name. Okay, so this is where the plot is being saved. So I'm adding an underscore JP here, executing this again. And so if I practice some patience, I suppose, then I will have this file pop up in my directory. Now I have genotype by biomass PNG and by biomass underscore JP. Showing you what I would do for exercise 1C. Welcome back to the Jupyter Hub. So we're in the copy of my notebook. And so one of our main tasks is we can add additional cells. That's true. We'll do this maybe at the very bottom. And we'll use Markdown and maybe write down results from long days. Execute this and add a new one. And so now we want to edit the code to depict the graph from the long days. Okay, so I'm scrolling back up to the plot that has been shared with me. I'm pasting it in here. So this is what filters for the short day data, but I want to use long day data, so I'm substituting LD. I'm also changing that in the title of the plot. And then I'm changing that name of the plot as well. Then I can hit run. What is going? Oh, this is the one. Apologies. Okay, so now I have my libraries loaded. And okay, so now I have the plot for the long days. We can write a figure legend for this too. So in the next chunk, I will use Markdown again. And I will take a quick glance at how they made the first figure legend, double click into the cell. And I'll actually copy their Markdown code and paste it in here and modify it to what I need. So this is long days. Okay. execute. And now we're adding our statistical testing. Again, I'm making a new cell for code. And I am going to adapt the code that has been provided to me for the statistical test. I'm changing the variable, I'm adding LD to it so as to not overwrite my previous results. And then I can execute this again. And finally, we want to look at these results as well. Fixing summary and executing to see my levels of significance. Okay, and then I could be interpreting these results. And here in this final chunk, I could write down my internal interpretation. Okay, so this would be it for exercise 1C. So now we can move on to exercise 1D, which is sharing your Jupyter notebook. And I think this is already in the pad. So this is our last exercise on this. So I encourage you to go through the steps, try to download your notebook as an HTML and a PDF, and look at the files, see how you like them, see what the difference is. If you encounter an error, let me know or write it down. And then fill in the questions here and kind of note the differences. I feel like that's somehow old syntax. So the error, I think, is caused by the image. So if you double click on this cell, you'll open up the markdown code for it. And I think you'll find that before the file name, it said attachment : and I found that if I removed that attachment : it actually worked to download. I think that's different markdown syntax. So I am in my notebook, and like I was just saying, I had to edit this one line here before it said attachment, and that didn't work for me. So remove this and then hit run again. And the way we save this as HTML or PDF is we go on File, Export Notebook As. And so the two options that we want to try is HTML and PDF. So for example, I take HTML and this opens an empty tab, but it does actually download my notebook. So I can click on this and see this as an HTML file here. Exactly. So, and this I executed without executing all of my code. So what you can see is that none of this code was actually run, and I don't see any output. However, if I go to run all cells, and then make my HTML, you'll find that it looks like this. So sure, I'll have some messages from R, but I can also see my data frame that I loaded in, as well as the images that I generated. So if I were to send this to you now, you could see my entire code as well as the output that I was seeing, that I was making interpretations on without running it yourself. Whereas if I just sent you this, you would probably be like, well, this is all nice, but I can't see your data. And similarly, if we can export this as a PDF as well. So we go to export notebook as PDF. And this takes a second, but it will download too. And so this is the generated PDF. You see the image is included. And we have the message here from loading. We have our data frame included. We have our plots. And so this is in a format that I feel comfortable sending my PI to, and like, here are my results from last week. And that's the end of our exercises. I've just one more slide. And I just wanted to point out that what I said earlier, that Jupyter Notebooks is not great for constructing really, really big analysis pipelines. It's great for doing small iterative data analysis, sharing your data, sharing your analyses. But if you find yourself doing huge pipelines over and over again, there might be different tools for this that are more robust and more efficient, can use parallel computing and maintain dependencies much better. And there are some examples like Nextflow, SnakeMake, and Galaxy that will help you achieve this probably much better than Jupyter Notebooks.